In the conference package, I sort of had titled this talk, Nutrigenomics for Obesity Prevention and Treatment. And I have to admit, that's admittedly very ambitious. And it's not exactly to the point of what I was going to talk about today. So I've retitled the talk a little bit to say, Predicting Changes in Body Weight in Response to Diet Using Transcriptomics. So I think it's actually very fitting for this session in the sense that we've now heard some work about proteomics, we've heard about genetics, and now we're going to switch to gene expression and transcriptomics. And again, I have to actually be honest, that saying predicting changes in body weight, that in itself is very ambitious. And I'm going to spend the next couple of slides to just sort of demonstrate just how ambitious a challenge this is to try to actually predict, successfully predict changes in body weight. So I think if we start off, I'm not going to surprise anybody by saying that obesity is a complex physiopathological state. And we tend to sort of refer to this in a typical sense as being obesity being a problem with energy intake versus energy expenditure. But that really oversimplifies things. And if you start to look a bit at the system that's underlying that premise, what we see is that there's a systems biology paradigm. Now I'm going to be focusing today on adipose tissue, so I quite like this slide because it's adipocentric. But with that said, we could actually change the positioning of these various organs to look at different organs and their impact and their contribution to obesity. And really, when we start talking about this imbalance between energy intake and energy expenditure, what we're really talking about is starting in the intestine, energy absorption, so the macronutrient composition that's coming from diet. That is being sort of distributed throughout the body, either in short-term storage in the liver or in longer-term storage in the adipose tissue, eventually to be utilized in the muscle, and all of this is under the control of the hypothalamus. And a lot of the molecules, whether it's cytokines, adipokines, gut hormones, and so forth, are being secreted from these various tissues and are integrated in the hypothalamus, and that's really what's regulating this balance between energy intake and energy expenditure. So I think when we're looking at a slide like this, we can say very quickly there's a systems complexity here underlying obesity. But then we can also add to that a developmental complexity. Obesity isn't just obesity. If you're looking at an individual walking down the street, two individuals who are obese, they might not be in that same developmental stage of obesity. So are they actually equivalent when we're starting to look at them at a molecular level? So we can ask that question when we're seeing this sort of curve, and there's purposely no numbers on the y-axis for weight or the x-axis for years, and that's because that's going to vary from person to person. But really what happens is there'll be a moment in an onset where there'll be a behavioral trigger or a genetic trigger or a lifestyle trigger that initiates a sort of gain in weight, and that will fluctuate a little bit as people are aware of this weight gain and the initial weight gain. They want to try to lose it, and it's going to be maybe very seasonal and so forth. Eventually you hit a point where that sort of plateaus. And underlying this initial increase here are a lot of gene-environment interactions. And I'll talk a little bit about the genes, I'll talk a little bit about the environment, what I mean by those two. But this is quite a complex state here. And what we get to here at this point where we decide either the individual or the clinician, it's time to make an intervention. And really that tends to coincide with the development of chronic diseases, or a permanent sort of inflammatory state, risk factors for diabetes, for cardiovascular disease. They start to appear quite significant, and it's time to act. And so this intervention here then can be a lifestyle intervention. It could be diet, it could be an exercise regime, or it could be something more severe like a gastric bypass surgery. In any case, the goal is to lose weight. And as we heard from Edwin just a little bit earlier, there's two outcomes here. One is we enter into that yo-yo state. So people actually who have lost weight then regain that lost weight and start going through this cyclical process. Or you can start to maintain that weight loss. And that's really very individual dependent. And that's one of the problems in trying to predict is what an individual, how they're going to respond to an intervention and how we can look forward to that. So I think basically now we can say there's a systems complexity, and now when we add this, we can say there's a developmental or an evolutionary complexity to obesity. But it doesn't stop there. We can then go to genetics, the genetic component. And if you look at this slide ruler, as you go from left to right, the idea is that the influence of genes is decreasing, for any one gene as environmental factors start to have more and more of an effect. So on the left-hand side here, what you have are monogenic obesities, very rare cases where you have one gene that is very predominant in influencing the obese phenotype. But as you go from left to right, as I said, that environmental influence is starting to take more and more of an influence, and that's really what we're talking about with everyday obesity or common obesity, or also referred to as polygenic obesity. And what we see quite rapidly is dealing with those different genetic forms of obesity, the actual treatment, there's different degrees of efficacy. So on top of a systems complexity and a developmental complexity, we can now add a genetic complexity. And when we're talking about genetics, we're actually in some ways oversimplifying things. We tend to think of SNPs first off, but there could be gene expression, there could be copy number variation. We've heard about epigenomics and epigenetics. That's a major factor that underlies this transition from lean to obese. However, all of this is happening in the context of an individual's environment. So we look at something like this, and these are just a handful of various environmental pressures that interact with an individual's genetic makeup to influence that progression and that severity from lean to obese. So at the very top here, what we see are nutrition and exercise, two of the major ones. But you see that there's things like viruses, social influences, psychological influences, microbial influences. All of these are new fields of research that are starting to show very significant contributions to the development of obesity. So to sum up really all these different aspects, we can definitely say that there is a systems complexity, a developmental complexity, a genetic complexity, and an environmental complexity. So now when I start talking about trying to predict changes in body weight, you can appreciate that this is a pretty significant challenge that's ahead of us. But nevertheless, I'm somebody that likes a good challenge, and I know there's a lot of you in the audience that like good challenges as well, and that's why we're all here. So if we're looking at trying to predict changes in body weight, well, I take an approach right now which I consider to be hypothesis generation. So we know that we have a population with an extremely variable phenotype, and we know underlying that there's genetic and environmental interactions. So you can use sort of approaches like genome-wide scans or gene associations, gene expression with transcriptomics, to identify candidate genes that may have an influence in sort of regulating that body weight. And then we can say, are those targets for a dietary intervention? So again, we're talking about polygenic obesity here. So what we know is that there's a number of different diets out there that are used to encourage weight loss, but we also know that there's a mixed efficacy. Some people lose weight, some people don't. It's a source of frustration for a lot of people. But that genetic component is nevertheless extremely important. And I think a study like this was published in 2000, gives a lot of power and a lot of encouragement to say that genotype does have a very significant effect on changes in body weight. So this is a study where they looked, the authors looked at 14 pairs of female twins, and they looked at changes in body weight and in fat loss. So on the left-hand side, you have weight loss, and on the right-hand side, you have fat loss. And this was a study where they did 28 days. The subjects consumed a very low-calorie diet. And if you look at either of these two plots, what each dot is representing is a twin pair. And if the two twins were losing exactly the same amount of weight or exactly the same degree of fat loss, then they would lie on that diagonal one-to-one line. But what you can quickly see here is actually they're not deviating too far away from that one-to-one line. So that it implies that there's very little difference between genetically identical twin pairs. However, you do see there's quite a spread, there's quite a range. Some of the twin pairs are losing smaller amounts of weight than others. So it implies that there's very little difference between twin pairs, but big differences between subjects. So in other words, some people will lose more weight than others on the same diet, but people with a similar genotype may respond in a similar way to a particular diet. So this is very encouraging to say that there might be a way of predicting this genotype to actually get an idea of the degree of weight loss or changes in body weight that might occur. So there's a number of different things that are being used right now to predict weight loss. And I've only pulled out a handful from the literature. There really are a large number of things that are being used. For the first example here, from a clinical perspective, just changes in weight loss. When people start up a weight loss program, that degree of weight loss that takes place in that first week can be a very good predictor of how much weight they're going to lose over the course of that intervention. Genetic polymorphisms. That's a rather unexplored field for the moment, I would say, with regards to changes in body weight, but it's certainly encouraging based on, for example, a polymorphism in UCP2, the uncoupling protein 2, that's associated with greater weight loss. And then finally, gene expression profiles. And this, in a sense, this study here was published in 2006, and it served as a bit of a basis or a bit of an encouragement for some of the research that I'm going to be showing in the next couple of slides. The idea here, what they found is, if you look at this particular image down here, each one of these curves, and this is probably very difficult from the back to see, but each one of these is a line that represents the weight evolution of a mouse on various diets. So there's low-fat diet, high-fat diet, and so forth. And what they found was that it doesn't really matter what you fed these animals. You consistently had mice that were high responders, so their body weight was going to consistently be higher no matter what you did to them. And there was mice that were low responders, no matter what you did to them. So this was very encouraging with the idea that we could maybe identify, using gene expression, whole gene expression studies, we could identify a pattern of genes that could be associated with high response or low response in terms of weight loss. So how did we do this? Well, Dominique Longin yesterday explained a little bit this, so I'm going to go very briefly over it. But basically, we're using an exploratory approach, or that whole genome approach. However, candidate genes, as we heard a little bit earlier, that idea of actually validating whole genome is another approach we can use to study adipose tissue biopsies. And all of the research that I'm going to show today is based on subcutaneous adipose tissue biopsies, quite simply because it's a lot easier to get than visceral fat. And this was an interesting finding. When we started to get the results of our initial weight loss study, and we started to explore a little bit the gene expression profiles, we said, well, we want to validate some of these changes. And we went back into our previously run microarray data sets, and we looked for validation, and we said, well, this is interesting. We can't actually validate some of the findings, and I'll present these findings a little bit later, but we couldn't validate them. So we went back a little bit further to look at the clinical aspects of how these actual biopsies were obtained. And this was sort of the underlying premise for this particular study that we did. And this was published in 2009. And what we did here is we actually compared the two different mechanisms, or the two different methods for obtaining subcutaneous biopsies. So one is the typical and the more commonly used needle aspirate, but individuals who are actually undergoing some sort of operation we can have access to surgical biopsies. Now, I do want to reassure, this is work that was done in France, and I just want to reassure everybody that to obtain surgical biopsies, they do not use a knife that looks like that. At least I've been assured of that. I'm not entirely sure, but what we have here is a 2x2 grid, if you will. And what we did is we obtained biopsies from lean individuals and obese individuals using both methods. And so you can see that in terms of body mass index, it was equivalent between the lean subjects, irrespective of how we obtained those biopsies, and age was similar all around. And so they were all sort of matched as best as possible. And when we looked at gene expression, so each dot here would represent the totality of an individual, really. This is a way of representing that in two-dimensional space. What you see very quickly is that those different biopsy methods and those different classifications, so whether it was a lean individual from which we obtained a surgical biopsy or a lean individual from which we obtained a needle aspirate, what you see is that they don't cluster together. So right away, we actually were able to see that there were differences in the underlying gene expression patterns that we were obtaining based on the biopsy method that was being used. So that explained quite a bit some of the difficulties we had when we were trying to validate some of the research and some of the findings, is actually when we went back, we realized that we were trying to compare needle aspirates versus surgical aspirates. And when we looked at this gene expression profile, we wanted to put some functionality into it. We don't particularly like looking at lists of thousands of genes. We try to make this a little bit more simple to look at. So we look at functional pathways. And when we compared obese versus lean, either using surgical biopsies on the left-hand side or needle aspirates on the right-hand side, the green arrows indicate functional pathways or functional annotations that were similar in both of those cases. So you can see there's very few green arrows that are actually similar between the two. But what we see is that there is on the left-hand side, these would be functional pathways that are upregulated in obese individuals, and on the right-hand side, those would be things that are downregulated in comparison to a lean individual. So we see that there's an increase in inflammatory pathways and a decrease in oxidative phosphorylation, things that we would have expected to see. But I want to bring your attention to a number of pathways in the surgical biopsies that really drove home the fact of what we expected to see. We wanted to see, and we were expecting to see, increases in inflammatory pathways, and sure enough, in the surgical biopsies, we have a large number of functional pathways related to inflammation and the immune response. And we wanted to see decreases in metabolism pathways, and sure enough, we see decreases in various metabolism pathways. Now, interestingly, as a functional classification, we didn't see those changes using the needle aspirates. Many of the genes, if you actually looked at the actual gene, there was a lot of similarities, but in terms of significant functional pathways, there was quite a difference. And we found that quite surprising, so we went and looked at the actual biopsies under a microscope. And if we look at the C and D first, it's a bit counterintuitive, but if we look at C and D first, C being a surgical biopsy under a microscope, D being a needle aspirate, this is we've zoomed in here on the adipocyte fraction. What you see are the adipocytes are quite intact in both fractions. We have the macrophage crowns that surround some of the adipocytes, so we see that in both those biopsies. However, when you look up and you zoom out and you look up at the surgical and needle biopsies above, you see that there's a significant difference. And this actually has to do with regards to the surgical biopsy method, being able to obtain this fibrotic region. So that's a new region that's been recently described in adipose tissue that's highly enriched in extracellular matrix genes and inflammatory profiles and so forth. So you can see that a needle biopsy does not have that fibrotic region coming out. So this was actually done, first of all, in different subjects. So we thought, well, maybe we didn't have any luck, and we actually identified a subject that didn't have any fibrosis in their adipose tissue. But we actually went back and took two different biopsies from the same individuals. We did this in a number of individuals, and sure enough, this held true. So a surgical biopsy, you're able to obtain this very rich inflammatory region, whereas the needle biopsy, you're not. So take-home message for this is really just to be wary about comparing data sets that are obtained using different biopsy methods. It's very difficult to compare, and you'll be probably pretty frustrated trying to do that. But now let's go on from this. So everything I'm going to be talking about are needle-aspirated biopsies and from subcutaneous adipose tissue. So can we use subcutaneous adipose gene expression profiles to predict changes in body weight? And as we heard a little bit earlier, this is obviously a major goal because successful weight loss and more importantly, successful weight loss maintenance is associated with numerous metabolic improvements. So improvements in insulin sensitivity, reductions in various blood lipids and risk factors for cardiovascular disease, reduction in inflammatory markers, etc. So this is a clear goal of why we want not only to encourage weight loss, but more importantly, to actually maintain that lost weight. And there's two papers really that have done this in a human intervention study. The first was one that was published in PLOS in 2007, and the second, hopefully Louis is okay with me talking a little bit about his paper. The second was in 2010. The first study, we used 53 female subjects, so 27 individuals that we found were considered responders. So these are individuals who lost during a weight loss period. They lost between 8 and 12 kilograms of body weight, and non-responders. So those are individuals who lost between 0 and 4 kilograms. So this was an intervention where what we did is we took biopsies at baseline, so time 0, and we wanted to see if we could predict whether they would be a responder or a non-responder in 10 weeks' time. And because this is actually quite a bioinformatically heavy prediction algorithm, there's a data set that's commonly used as a positive control, if you will, to make sure that our algorithms are working correctly. And this is called the Golub cancer data set, where they looked at individuals who had acute lymphoid leukemia and acute myeloid leukemia. And if you look at these two plots, this would be our plot here of weight response. Again, that idea that each dot represents an individual. The red represents non-responders, the black represents responders. If you compare these two plots, you see instantly there's quite a big difference. that when you're looking at the leukemia subjects, there's a clear separation between the ALL subjects and the AML subjects. However, if you're looking at our data here, we're looking at responders and non-responders, there's a considerable overlap, but there is a tendency to separate. So we thought this was actually encouraging enough to go forward and see if we could predict changes in body weight. Now, I'm not going to get too much into the bioinformatics and the algorithms that were used, but needless to say, what we do is we basically do iterations where we create a test data set, or sorry, a training data set. So that's where the model knows whether an individual is a responder or a non-responder. And we create an algorithm, and then we go and test it in a subset of subjects that we've kept aside for the moment, and we go and see how accurate we are in our prediction of whether they're a responder or a non-responder. And you can do this with any sort of number of different genes that are used, but the goal really is you want to be able to predict better than flipping a coin. So if you can't predict very well, you're going to get a 50% chance. Heads or tails, the person's going to be a responder or not responder. So we want to make sure that our algorithm is doing better than that. And of course, like I said, we're using this control cancer data set to sort of validate our algorithms. If you look down here, this bar down here, this is the cancer data set, and I'm sure you can't see those numbers in the back, but our algorithm is working very well because we're predicting with about 95–98% accuracy whether an individual is going to have acute lymphoid leukemia or acute myeloid leukemia. Now, if you compare this to our data set, basically we didn't need to do gene expression. We could have been flipping a coin and we would have had about the same accuracy. So we have about a 50% accuracy here, which is not very impressive. So at this time, we were basically concluding that a single gene expression snapshot prior to a dietary intervention, so at baseline, is actually insufficient to accurately predict weight loss. Now, we've continued on with this sort of research and changed very significantly how we're approaching our groupings. And so we've actually made some improvements now that are it's a manuscript in preparation. But I'm not going to talk too much about that. However, I am going to talk a little bit, just briefly in one slide, about the second paper here by Louis and his colleagues, which they basically found the same thing, that if they took baseline gene expression data, they found that there was no way of differentiating individuals who are going to become high weight loss responders and low weight loss responders. Now, there's a significant difference, actually, between how they classified their subjects. We looked at actually kilograms body weight. They looked at percentage in terms of initial body weight loss. Nevertheless, the conclusion is the same. The gene expression profiles at baseline don't necessarily, can't be used for predicting accurately whether an individual is going to lose weight or not. Now, interestingly, this study here, they identified some changes in the epigenetic profile in genes related to weight control and insulin secretion. So it's actually quite encouraging to think that, yes, gene expression might not be sufficient, and I certainly would not go out and say it's definitely categorically not going to work. However, at this time, it's not working. But I would say that the epigenomic profile may prove to be more informative with regards to predicting changes in body weight. We then went on and said, like I said, we are easily discouraged, so we continued on with this line of analysis, and we said, well, can we predict successful weight maintenance? So now we've switched from saying predicting weight loss to actually predicting changes in body weight that are maintained in over a 6-month period. And the first study came out a little bit earlier this year, and I'm not going to talk too much about that because Dominique Langin spoke about that yesterday. And I'll focus more on the second paper here that's currently under review. But just to sort of set the context, this was the framework, European Framework Project Diogenes. And it was a study that took place over a period of about 8 months, the actual intervention. There was a 2-month low-calorie intervention, followed by a 6-month weight maintenance period. And what we actually were interested in doing was looking to see individuals who are able to maintain lost body weight. So we're interested in this 6-month period here, but we wanted to know, do they actually respond to a low-calorie diet differently than people who regain weight? So we're interested in knowing changes in body weight here, but we're actually looking at changes in gene expression during this low-calorie diet intervention stage. So can we use that to predict successful weight maintenance? So in this particular study, we had 40 individuals. Each one of these dotted lines represents a different individual, where we classified individuals as weight maintainers and weight regainers. So a weight maintainer was an individual whose weight fluctuated less than 10% of their, less than 10% of their body weight was regained during that 6-month period. And weight regainers were individuals who regained between 50 or even all the way up to 100%, even more than 100% in some cases. But what you can see when you look at the groups together, that in terms of the weight loss, they're actually very matched. So when we looked at common bioclinical parameters, things like BMI, fat loss, changes in insulin sensitivity, and so forth, they're actually very similar between the two groups. So we made sure that they were matched between these two groups. But that didn't mean that the gene expression profiles wouldn't be different. And sure enough, we found that there are actually fairly significant changes in gene expression between weight maintainers and weight regainers during that low-calorie diet intervention. Now, this sort of drives home the point sometimes that looking at individual genes, it's a bit difficult to get an appreciation of what's happening at a functional level. Because you can see that there's a number of genes here, over 1,000 genes changed in weight maintainers uniquely, and 1,000 genes changed in weight regainers uniquely. But when you actually look at their functional profiles, they were very similar. So it means that there were genes and similar pathways that were being affected, but when we looked at those two groups, it wasn't particularly the same genes per se that were being changed. But functionally, we would say that actually there's very similar response. But however, there were two categories here that we found particularly interesting. The first were genes that were in directional concordance. So that means if they were upregulated during the low-calorie diet in a weight maintainer, they were also upregulated in a weight regainer. Or conversely, they were both downregulated. And here we found there were some very strong similarities with regards to fatty acid metabolism in particular, specifically polyunsaturated fatty acid metabolism. And we were very interested in this, what we call the oppositely regulated genes. So that means these were genes that in a weight maintainer were downregulated during the low-calorie diet intervention, but were upregulated in the weight regainers. So they had a different sort of profile during that low-calorie diet intervention. And what we found to be particularly interesting here was the fact that weight regainers had a very significant upregulation in extracellular matrix functionality. And the weight maintainers actually had an increase in apoptotic pathways. So this, as a hypothesis-generating approach, proved to be quite interesting. Could we suppose that actually weight maintainers are experiencing in adipose tissue an increase in apoptosis that then would be underlying potentially that effect in successful maintenance of weight loss? We went a step further, and this is a very pretty and very complicated figure to look at, but what it is taken that gene expression, and we've basically associated an enzyme with each one of those, a protein or an enzyme with each one of those genes, and then we associate that using a metabolite reconstruction analysis. We then associate those changes in proteins with potential changes in metabolism. And really the take-home message here, so if you're looking at these diamonds here, red would be an upregulated gene, green diamonds would be a downregulated gene, and these blue circles here, or these turquoise circles, represent the metabolites. And this is what the metabolite reconstruction looked like in weight maintainers, a very highly interconnected pathway or underlying network. But now if you look at the weight regainers, very, very disjointed. That sort of connectivity that we saw with the weight maintainers just simply doesn't happen with the weight regainers. So it's really showing that there is a very significant and very unique response to a low-calorie diet that differs between people who maintain their weight loss and people who regain their weight loss. So to sort of take-home messages for this, and I didn't really talk too much about this because Dominique Langin mentioned this yesterday, but definitely changes in body weight affected adipose tissue gene expression far more significantly than changes in macronutrient content. But what we could clearly say is that weight maintainers and weight regainers do not respond to a low-calorie diet in the same manner. The weight maintainers experience a highly coordinated and interconnected response to a low-calorie diet, as you saw by that metabolite reconstruction analysis. Weight maintainers have significant increases in genes related to apoptosis, fatty acid metabolism, and oxidative phosphorylation. So some of these groups or functions were also seen in weight regainers, but however, the one I want to underline is the apoptotic pathways. We saw changes in caspase genes. And as you clearly saw, weight regainers simply do not have the same sort of response, either from a gene expression level or from a metabolite reconstruction pathway. They'd have a very disjointed and disconnected response to a low-calorie diet. So just to sort of wrap up here, some of the challenges that omics-based research faces with regards to prediction. I think this is at the very beginning. It's very exciting, the stuff that's happening. We're learning and we're moving forward with this. As you can see, bioinformatics plays a major role in all of this type of research. And so getting accustomed and understanding how those bioinformatics algorithms works are clearly important. But as I mentioned at the very beginning, what you see right off the bat is that obesity is a complex phenotype. We're trying to predict something that has a system, developmental, genetic, and environmental complexity. Not an easy task. We also know all those various differences certainly contribute to the inter-individual differences that we see. And this just a few things like genetics, lifestyle, exercise, and so forth. Those are factors that will have a major influence on trying to successfully predict changes in weight loss. And we can also ask the question, I take this from a gene expression perspective, but there's nothing to say that we couldn't be doing the exact same thing using either genetic polymorphisms or plasma peptides or proteins or metabolites. All of these will provide different quality or different types of information that actually may prove to be more successful with regards to predicting changes in body weight. We can ask the question, and I think this is a very relevant question, are omic or genomic, transcriptomic, so forth, are those snapshots sufficient? Do we need to have more of a dynamic situation where we take repeated measurements and look at how an individual is responding to a change in diet to actually be able to better predict long-term changes? Clearly, there's advantages and disadvantages to this point, but that's an important point to consider, I think, in future research. And then, like I mentioned, bioinformatics. We can also ask the question, comparing, when we were looking at, for example, that data set that was looking at leukemia, whether it was acute myeloid leukemia or acute lymphoid leukemia, we can ask the question, are dietary patterns and changes, and minor changes in response to dietary influences, are the current bioinformatic analytical tools, are they actually sufficient to actually be able to determine small, minor, subtle changes in gene expression versus, for example, 2 different types of diseases? So we might be actually using informatic tools that are well suited for a certain situation, but maybe not ideally tuned for another. And with that, I'll wrap up. I'd just like to thank my colleagues at INSERM Paris, where this work began. This was all in the context of the Diogenes project. And since my move to Canada, I'd like to thank, of course, the various organizations that allowed me to continue on this research. Thank you very much.